Hello everyone. So today I'm going to be guiding you through our SpotMap MS LCMS analysis software, which is designed for the automation of the process of analyzing LCMS traces for the presence and for quantification of host cell proteins. Now, this is designed to reduce interoperator variability through automation, and it's designed to save a huge amount of time for large pharmaceutical companies, CDMOs, service providers, those kind of things, against existing tools. Now, all of the software within this space is based on LCMS, basically proteomics software. It isn't designed specifically for host cell protein analysis, and we believe from TotalLab that is one of the problems with it. Basically, it's not designed with a clear focus, so you end up with a lot of data at the end of your LCMS analysis, and then it's up to you, the operator, the analyst, to wade through all of that data and try and work out what's a host cell protein, try and perform your quantification, and try and get the answers that you're looking for. Usually, typically, that answer is, do I have any host cell proteins that are a known threat to either humans or the recipient of my drug, or anything that's going to degrade the drug, the product, when it's in storage, so reduce the shelf life and increase the cost to consumers of that drug. By using SpotMap MS and having a very quick readout of the presence or absence or even quantity of host cell proteins and categorizing them by threat, operators get a much quicker answer to the questions that they're asking of their drug substance. But it can also be used for, biotherapeutic purity assessment. So to check if there's any common contaminants from your generation process of your new therapeutics. It's really got quite wide-ranging applications, and we're going to build on that with a proteomics piece of software in a couple of years' time. So I'm going to show you through how we set up an experiment and through the automation steps, because I think for people coming from other pieces of software like Proteinmetrics, Diane, Skyline, whatever you may be using, are going to really appreciate how much variability we've taken out of this assessment. So typically what you would do is you would run through your system setup. So you would add your name, your company name, and the name of your mass spec device that you're using. So if you've got multiple, you can give them nicknames and have, be able to read back from your PDF reports, your results, to see which machine all of your results came from. Likewise, you can have your username and your company name show up on your reports. So if you're a service provider, you can generate kind of customized reports that go to your customers to say, this was performed by this person at my company for your company. So these are somewhat compliance features, but we do have full 21 CFR and GMP compliance. I'm not going to go into that at this stage, but just to let you know, we have that as an option if you're working in GMP or 21 CFR requirement your spaces. Next is the protocol setup. So you can see here that the software comes with a built-in FASTA file and HCP database, but you can provide your own FASTA files in standardized format. And what the software is going to do at kind of a high level is when you add your traces in, it's going to search your FASTA file and search the HCP database that we've provided with the software and use that to see if there's a match in any of your traces. And then it's going to present you kind of the risk factor, the risk level, and where that risk lies for the host cell proteins that are detected. So it's as simple as provide a FASTA file, provide your raw traces from your mass spec, wait for the process to happen, and you will get a report that shows you what's in your sample. And you'll see that as we move through the demo. You can have saved protocols, so you can have templates. So you can import and export your protocol. So if you want to do any kind of tech transfer, if you're a service provider and a user has provided you with their protocol, you can be guaranteed that you're running the exact same settings, you're doing the analysis in the exact same way. So this is one of the features we've introduced to reduce subjectivity using this technique, allow tech transfer, allow harmonization between maybe different sites located worldwide to make sure your pharma company X, is doing everything the same way in all of its different locations. So there's a certain measure of standardization there. You can turn on or off common contaminants. That's things like, if there's any trypsin left over from your digest, things like that. That's an additional. So the common contaminants are not host cell proteins per se, but they may be signs of impurity. The purification steps have not been successful, and you might want to include that in your results for your process optimization step. If you want to normalize to a therapeutic protein, so if you want to perform quantitation of host cell proteins relative to your therapeutic, you can add the accession in here. You can also include spike proteins and include the concentration of your spike proteins, again, for quantitation. So we don't perform absolute quantification within software, we perform relative quantification, and we represent that as equant within our results, and you'll see that shortly. The reason we do it that way is because there is a lot of debate within the host cell protein quantification world via LCMS, and really the only way to do this accurately is to say that it's relative quantification rather than absolute quantification. That it is possible, but it's it has a lot of disadvantages. It has a lot of caveats that I'm not going to go into here, but for most users, relative quantification, how much host cell protein is there relative to my drug is what most people are looking for. And here at the bottom we have the host cell protein database, which is basically our lookup and contains all of the information about the kind of threat level assessment of the host cell proteins that the software has found. We provide one with the software. It is AI generated, and it uses AI to scrape all of the available publications based on host cell protein where these things have been reported. We collect them all together and we use that as our host cell protein database. But if you watching this, there's a lot of pharmaceutical companies that have built their own host cell protein database that are specific to their workflow, things that they know might come up every now and then based on their cell lines, things like that. We can work with you to integrate that as well. So please reach out to us if you've got a large database and you want to be able to kind of automate your process like this. Definitely something we can do, but for people that are just starting out, we do provide that information as well. So this, I think, has about 51 host cell proteins in it with threat level assessment. So yeah, it's fairly comprehensive, but of course we can't guarantee that it contains every host cell protein that will ever exist. So to create a new experiment, I'm not going to create a new experiment. I'm going to just open an existing experiment because it's going to be much quicker to open an existing experiment than it is to analyze a full new experiment. And it wouldn't make for the world's most entertaining video for you, the user, the watcher. So we would click this button here and open up a window to bring in our raw trace files from our LCMS. We've got a summary of all of our different, our FASTA file that we're using, if we've added any therapeutic or spike proteins, which experiment database we're using. We've got kind of a summary of those there, and we can give it an experiment name and again a client name for the same reasons that we allow you to kind of put a company name on your report. So this is where you would name the experiment and name the client if you were a service provider, and all of that information comes out on the PDF report at the end. So you've got a really lovely, documentation that you can send to your customer and say, here's how we performed your experiment, these are your results, here's your quants, and it looks really professional. So watch mode is a way of monitoring a folder and processing LCMS files, raw traces, as soon as they come into that folder. So you can see here your current experimental parameters that we've just set up before. The idea is as soon as a raw trace hits the folder that the software is watching, it runs it against these settings and it just goes straight away. So you can do things like, if you're running an experiment and the files only come out at the end of the day, if you set watch mode up, you don't have to stay an extra 2 or 3 hours on the end of your day as a scientist to be able to process those and wait for it all to be done. You can leave safe in the knowledge that when you arrive in the morning the next day, the software will have actually processed all of your data for you, and you can get through to the kind of important step of making decisions about which matches are true and look through the quality of the data and things like that. So it makes the analysts' lives a lot easier, fewer late nights, fewer weekends worked, things like that. De-stresses and speeds up the whole process of doing this via LCMS. And then obviously open existing experiment is what I'm going to do right now. Opens a previous experiment. So what you're seeing here now is the software is processing the experiment that I just opened. Now this is quicker than it would be with a raw trace because this is already a previously analyzed experiment. You can see how quick that was for a triplicate sample. So it's still processing here, but we don't wait for it to be fully processed before we start showing you some data. So the first thing we show you is the visual QC readout. I've got a separate video on this, on what the visual QC is for. But it's a very quick QC check for the operator to see if their sample has run correctly. So these are triplicates. You would expect them to look very similar as they do. There's some variation, of course, as there are in all things in biology. There's some variation, but they look largely the same. The visual QC is great for spotting equipment failures or failures in samples, things like that. You know, if things don't all look the same, it allows you to very quickly rectify that. So whether you need to run a sample again, you need to, load a new column, something's gone wrong with your equipment, you get that information extremely quickly, which means you don't have to wait till the end where you've processed all of your data to be able to intervene and kind of save that time and make sure that your experiments are running correctly. An example that I go through in the other video is we received a set of data from a collaborator when we were working on this piece of software, and you could see 2 out of 3 of the traces were fine, but one of them was abruptly terminated here. It was very obvious that something had gone wrong during this run, and when we reported this back to our collaborator and showed them this screen, they said, oh, well, we wouldn't have known about that until we had processed all of the data, got to the end, and our numbers didn't make sense. So for them, our software versus what they were using at the time would have saved them a huge amount of time and alerted them to bad data much quicker. The next section that the software comes to once it's finished, and you'll have seen me click off this quickly before, once it's finished processing, it takes you straight to the result, the report. And you can see here, this is very visual, but we've tried to include all of the most important data straight away on this page. So we've got our 3 traces at the top, and we've shown all of our, MS1, MS2 data, our relative quantities. We don't really have relative quant because I didn't add any therapeutic protein or peptide. Sequence coverage, and these columns here are actually a visual representation of the measurements from each trace. So you can see here, a lot of people, this is a great example, a lot of operators that we spoke to would reject a sample like this where it kind of shows only in 1 out of 3 replicates. Most people want to see, a presence in 2 out of 3 traces that they run. It's why they run them in triplicate or even greater numbers. They want to see consistency, so they would be able to see straight away this is suspicious. But the rest of the columns stay fairly similar across the different triplicates. You can see that there again. So from here, you're already getting a sense of the quality of your data and the quality of these matches. And based on the relative quant, what is the concentration of these host cell proteins that is detected. So we've got this system whereby we kind of rank them based on color. We can see straight away we found 752 HCPs. 5 are potentially high risk, and we can see the risk category here. So 3 of those that have been detected can degrade your drug quality. So they might be lipases that's going to break down, the solution that you've got your drug substance in for stability. Could change your shelf life from 6 months to 3 months, 3 months to 2 days, something like that, in really minute quantities. So even though here we can see it's a very minute quantity, it has been detected. But again, when it comes to kind of the manual step, you might, as the operator, choose to exclude that, and you absolutely can do that. But straight away, we kind of flag it up like, hey, there might be something here. Immunogenicity is exactly what it sounds like. It means this host cell protein has been reported to potentially have an immunogenic effect when administered to humans. So again, this falls under a high risk, but then a different category of high risk. So it depends on kind of what is your sensitivity to risk within your company. Are you happy with things that, you can work very hard to try and get rid of all host cell proteins, but actually do you just want to get rid of the ones that are immunogenic, and you will try and work around the ones that affect drug quality, or are you really focused on drug quality and not immunogenicity? Depends on the therapeutic that you're making, the dosage profile of that. as to whether that can be kind of a measured risk, but risk in this sector is a different topic altogether. So this is what you would see, and from here you can just actually make a lot of decisions and a lot of assumptions without having to process all of your data. But if you do want to run through your data with a fine-tooth comb, we've got lots of different quality control measures. So we've got the quality control step here, shows you the total ion chromatograms laid over each other. And we can see a lot of our information here. Again, we're using the 3-column system to be able to highlight similarity between different sections, because displaying kind of some of MS1 intensities like this, the scale of the numbers involved make it very difficult for people to get a scale of relative difference. So there is a difference here of approximately 500,000. There's a difference between this number and this number of, is that 500,000? Again, we see exactly the problem here. I think 500 million, I think there's a difference here of a billion of the MS1 intensities. But you see the point. It's much easier to look at the values like this relative to each other to see changes. This one's a very significant difference here. But it's a much more visual way of getting all the information that you, the operator, might need to check the quality of your samples. We've also got a full visual view of our RT, of our, just reading that, of our elution profiles. So have our proteins all coming out, have our peptides all come out at the same time. We can turn on or off closely eluting peptides. So if I'm interested in looking at this peptide here, what else came out at roughly the same time, I can have a look at that. And then I can come down to peptide inspect mode, which is kind of the human in the loop section of the analysis. So it's where you can say, actually, I don't believe that match is good enough quality. I'm not going to include it in my report. So we do have this human intervention steps to be able to allow operators to make the final decision, the final call on what is included and what is not. We can include the peptide steps here, but it's hidden by default. If you're using a larger monitor than I am, you would obviously get a better view of these sections here. I'm using a laptop, quite a small screen, quite low resolution, but this is software designed for 4K resolution, so we would always recommend people using that. We're trying to get as much information on the screen as possible for users to make these decisions and make good decisions, who are armed with all the knowledge that they can get from their experiment. But that obviously necessitates using a lot of screen space, but we have tried to optimize for that. It's just not optimized for this particular laptop, but 4K screens are very accessible these days, so that shouldn't be a problem. So what we can do here is we can move through the list using our keyboard and pass and fail different peptide matches based on this. So the idea is that when a user becomes proficient enough with the software, they can simply look at the screen, look at the graph showing, MS1, MS2, the different intensities, and based on that information, they can either pass or fail the peptide. So if they come through and they pass these matches, you can see as I'm doing it, kind of go, yep, no, that's dodgy, I don't believe in that kind of thing. As I'm doing it up here, it's color coding the kind of all of the peptides that have been detected or a match has been derived from, and it's building up a color chart to basically say how confident we are in the overall assessment of this trace. So we've got our 3 traces here, and we can run through all this manually to say, yes, I believe that peptide match is correct, so this protein assignment is correct, or we can reject it. And by looking at these columns, we can see, if 75% of it rejected, we're going to reject that, those results. But if most of it is green, we know that actually the vast majority of peptide matches for the proteins that have been identified are true, are positive, based on whoever has been, reviewing this data and their names on it. So you can always check back and see who's done this work, etc. Then once we get to the reporting section, we actually keep that color-coded, column view. So when you're giving a report to someone, whether it's an internal report or it's an external report, we can view the report and show the different columns. So now in our export and our report, you can see that we've carried through this color coding for the peptide matches that have been verified by a person, by an analyst. So by taking this approach, we've combined all the benefits of automation of kind of checking our matches and things like that, but we have given this step whereby a human is involved, because the results of this are incredibly important to drug safety and drug stability. We still have humans being able to be in the loop, be part of the validation and verification process. So it's not completely automated, and that's by design for safety. But if you're a service provider, when your client, your customer, or when you're distributing these things internally for a project, you can see very quickly for each of the traces, if they're fully green, it's a great run. All the peptides have matched up properly, all the qualities is great. But we can see very quickly using this color coding scheme, and again using these columns that represent each single trace that have been through in triplicate. It's a very good way to pass that data on to someone else for them to review and have a look at. And it also is a great way for you to go and look back at historic experiments, 5, 10 years in the future. You may not be able to remember, oh, what happened, what did we do, how did it go, was that run good or bad? If you just look up the report, load up the report for that experiment, you'll get a very good visual indication. So by using the SpotMap MS software, we're able to massively reduce the stress of performing this work, massively reduce the time taken to perform host cell protein analysis by LCMS as part of your advanced therapeutics workflow. It means that if you're a service provider, you can perform many more services. So with investment in this software, but without investment in additional staff or additional, mass spec equipment, can massively increase the throughput of your experiments, the throughput of your work, to be able to bring on more clients without having to worry about bandwidth. But yeah, you can de-stress your workforce, your people doing this. If you've only got a small team doing this, and they've got say 90 projects to run through in a quarter, they're going to be able to do that much easier and much quicker by using our software than the currently available tools. But don't take my word for it. Please head on to our website, get a free trial of the software, try it out with your data, see how much time it could have saved you if you had it when you were doing your last experiment. I'll include a link to the free trial for the software in the comments below. If you've got any questions or any queries, please feel free to reach out to us. We can set up a call, run through a demo, and speak to you about your process and how our software can help out with that.